Now we can see about the structure of the cochlea in detail. Okay, so this is a cochlea. The cochlea is a structure, it is a short coil tube. So this is a structure that helps in the hearing of the birds. Okay, so it is having a complicated structure. So each cochlea, it has three chambers. The upper chamber, it is called as a scala vestibuli. The middle chamber, this is called as a scala media, and the lower chamber, this is called as a scala tympani. Now the upper chamber, it is separated from the middle chamber, that is scala vestibuli. It is separated from the scala media by means of a membrane, and this membrane is called as a Reisner's membrane. Now the middle chamber, scala media, it is separated from the lower chamber, scala tympani, by means of another membrane, and this membrane is called as a basilar membrane. Okay. So these two membranes divide the entire cochlear chamber into three separate cavity: upper scala vestibuli, then middle scala media, and the lower scala tympani. So this diagram will give you a much more concrete idea about this. So this is a cochlear duct which has been enlarged to, I have drawn the diagram in this manner, so it will be better for you to understand. So this is the upper chamber called as the scala vestibuli. This is a middle chamber called as the scala media, and this is a lower chamber called as the scala tympani. The scala vestibuli, it is separated from the scala media by means of the Reisner's membrane. Now scala media, it is separated from the scala tympani by means of another membrane, and this membrane is called as a basilar membrane. Now the scala vestibuli and the scala tympani, it communicates with each other at the tip, at the posterior tip, and this by means of a small opening, and this is called as a helicotrema. Helicotrema, through which the scala vestibuli communicates with the scala tympani. Now the scala vestibuli and the scala tympani, they are filled with the perilymph, while the scala media, it is filled with the endolymph. Now the opening of the scala vestibuli, it is a round, oval opening. So this is the oval window that we have mentioned earlier, that the stapes will come and touch with the membrane that cover this oval window. Now the opening of this scala tympani, it is round, so this is called as a round window. Okay, so we have already mentioned this earlier. Now this scala media, it is separated from the scala tympani by means of the basilar membrane. Now this on this basilar membrane are located several sensory cells, columnar cells with hair. So sensory hair cells are located on the basilar membrane. Now this is called as the organ of Corti. So this has a definite role in the process of hearing. So on the basilar membrane, above the basilar membrane, several columnar sensory hair cells are there. So that will be helping in the hearing. Okay, and now above this organ of Corti, or the sensory hair cells, from this a posterior end, there is a small membranous flap which just located just above the sensory hair cells without touching them. Now this membrane, this is called as a tectorial membrane. Okay, tectorial membrane. So these sensory hair cells, along with this tectorial membrane, it helps in the hearing. So this is called as the structure, combinedly called as the organ of Corti, which helps in hearing. So this is a structure of the cochlea. Now when we come to speak about the mechanism of hearing, the sound, it comes through the auditory opening, through the auditory canal, it touches the membrane, tympanic membrane. Now it starts to vibrate. So through these skeleton rod like structures, the extrastapedial, suprastapedial and infrastapedial, the vibrations will be passing to stapes. Now the stapes, it will be touching, in as I already mentioned, it will be touching the oval window, it will be touching the membrane of the oval window. Now when the oval window vibrates, you know this tube, it is filled with the fluid called as a perilymph. Now this perilymph, it will vibrate, and through helicotremma the vibrations will be passing to the scala tympani. Now this fluid also vibrate. Now this basilar membrane which floats, it will be also vibrating. So when this vibrate, these sensory hair cells, they will touch the tectorial membrane. So now the touching of the sensory hair cells to the tectorial membrane, it causes the stimulation that will be passed on to the brain through the auditory nerve. So that is a mechanism of hearing in the case of the pigeon.